Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 131st seminar, and I call it a Zhejiang University Day. It is already August 1st. Only 5 months remain this year. We had 3 weeks of summer break, but we will not have any break for this weekly seminar series until Thanksgiving Day in US. As you may know, we will have the first in-person SynBYSS conference in Hawaii from December 9 to 12, finishing this year with the conference and the last webinar on December 12 as well. Actually, our official website for the conference actually released today with the registration and abstract site. So please visit the JCVI SynBYSS website for consideration of your abstract submission, and also joining the conference in person for the first time. I had a great time meeting many colleagues and friends in Dublin, Ireland during the 3 weeks break, traveling almost 2 weeks in Europe and West Coast. Importantly, I finally moved to San Diego physically, celebrating my official joining J. Craig Venter Institute, which has been made on paper on May 1st. There will be at least 4 more conferences in San Diego this year, including AICHE. And if you are in San Diego, please let me know so that I can arrange a seminar for you, or even a mini symposium for you, or if the multiple people are in San Diego, especially during or after or before the AICHE week. All right, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Baojun Wang. He is currently a professor at Zhejiang University. Before joining this university in China, he was a professor in UK. Actually, I also heard he's right now in UK attending a conference. He was in UK for many, many years as a professor. I have known him since 2011, I believe, in the SB5 conference at Stanford University, where he was presenting a poster on genetic endgate. And of course, at the same time, I was presenting my 4-input endgate work as a postdoc at the Chris Voigt lab at UCSF before we just moved to MIT. I believe we have intellectually interacted with each other since the first meeting in Stanford, and developing genetic sensors, circuits, and Baojun is world-renowned expert in genetic circuit and sensor building and analysis. Baojun, thanks so much for your great contribution to the scientific community, and also for agreeing to giving a talk. I thought originally very inconvenient time, China time, 11 p.m., but I'm glad you are in UK, much better time zone right now. And the virtual podium is actually all yours now. Thank you.
Okay, thank you, Taeshuk, for inviting me to speak here today. I think it's a great joy and honor being here to exchange any ideas with you guys. And also choose this specific date because Lauren is a speaker also choose this date because we have shared interest on the genetic circuit design. Yeah, Great, that's the reason why I choose this date. Okay, now I'm going to share my screen to start my 5 minutes talk. Um, can you all hear? Can you all see my slides now?
Perfect. Go ahead.
Okay, great. Okay, so today I'm going to briefly introduce a new paradigm or a new way for genetic logic circuit design. Because I have been working in this field since my PhD, which was since I joined Imperial College in 2007. I started working in the field of synthetic biology, especially on genetic logic circuit design. As Professor Moon mentioned, we actually met in 2011, and we have a shared interest on genetic logic uh, and get design in that early time. And I think today we'll continue this conversation. So one of the key aim of synthetic biology is to aim to design and construct new biological entities like proteins, genetic circuits, and cells, or redesign of existing biological systems. And I think the key concept or the difference that separates synthetic biology from traditional genetic engineering is that it is extensively use engineering principles and to engineering these biological systems. As you can see from this picture, engineering system, we have this from bottom up construction from materials to transistors, logic gates, and circuits and computers, etc. But in synthetic biology system, at the beginning we also share the same kind of philosophy. We want to use bottom up constructing to construct from very simple building blocks from DNA to biological parts like these different promoters, etc., to logic gates or others, and then to more complex genetic circuits, and even finally to cells and tissues here. But then I think at the beginning, the engineering principles and helps a lot to construct these simple genetic devices and circuits here. But then gradually with the scales to become increasingly large and complex, and we found that if we just simply adopting the engineering principles, and the challenge will be huge, because engineering systems can use even billions of logic gates or even billions here in one systems. But biological system do we need or can we do that? Because for example, in bacteria, we only have about 4,000 genes. In human cells, we are in about around 20,000 genes, but we can do so many complex, amazing things. So I think that one of the potential development for synthetic biology or for circuit design, that we need to balance the complexity and efficiency. How we can using this less amount of genes or less amount of genetic content to do more complex things, instead of just to expand the complexity of circuit design. Because in biology system, especially in cells, we don't have the same like insulation as in physical systems. So we need to think up a different strategy. So that's come to topic of my talk today, and the way I would like to share is that there's one way that I propose that we can scaling up genetic logic circuit design by splitting biomolecules. So the split molecules, in terms of those two ways we can do that, is one is at post-translationally, we can use split inteins that can split proteins into two halves and religate them to reconstitute this function. And scarlessly, for example, we can split a transcriptional activator like ECF extra cytoplasmic function, ECF sigma factors, and then after each half has been separately expressed from the two independent promoters, and they can through the protein transsplicing, and the intact ECF can be reconstituted to active downstream promoter. So this becomes the ANIGate. And similarly, if we also in the post-transcription, you also can split the RNA molecules using split introns. For example, group 1 introns, you can split mRNA molecules and religate them together into intact RNA molecule to produce a functional protein. For example, like a GFP protein. So in this case, we can only can split at RNA level, we also can generate a logic AND gate for the ECF transcriptional activity proteins here. So by doing this, it means that we can, using one protein, we can do more complex like logic gate designs here. And most importantly, we have many orthogonal, potentially orthogonal split inteins and split introns available. And we are already, in 2020, we developed an expanded library of 15 orthogonal split inteins. And recently, we have a publication just accepted, and we also engineering a large library of orthogonal split introns that can split any RNA molecules in different locations. And just give example that we can design a 4-input genetic logic gates by splitting at RNA levels and also at protein levels. Let's take the ECF protein like ECF20, ECF sigma factor as an example. We can, using, for example, design a 4-input AND gate. We can, RNA levels, we have 4 different RNA inputs, and the first 2 RNA, they can be religated through a split intron, we generate RNA and can produce ECF20 intin half, yeah, and another 2 half can generate, another ECF20C intin C halves, yeah. And then we can use the split intin that can reconstruct ECF20. By doing this, we can just using one transcription factor to construct a 4-input logic gate. And you can see the data, the dynamic range is very high. And similarly, we also can split a transcription repressor. For example, this case is a LMRA repressor protein, which is a tetrafamily protein. We can generate a 4-input NAND gate. I know Mozart did a pioneering work in this field. Back in 2012, he published a Nature paper with a 4-input AND gate that can. Now this is a layered 4-input AND gate by layering the 3 2-input AND gate into this 4-input AND gate. So together, this system uses 6 transcription factors to achieve this. However, in this system, we only use one transcription factor, but split at RNA and protein levels, use different orthogonal split introns and intins here. So we can, in this case, we can using one intin instead of 6 one transcription factor instead of 6 to do this. So that means we can increase the signal processing capacity of a single transcription factor by 6-fold. Similarly, we can use this protein to build more complex genetic logic gates. For example, here is a 6-input logic AND gate we design, and here now we can split our RNA levels into 6 pieces here. There are 6 inputs, and we can use 3 orthogonal split introns to reconstitute each 2 of them into these halves. Yeah, and now this is and the RNA levels and then produced into proteins, and at the post translation level, we use 2 orthogonal intins to reconstitute the final ECF20 transcription factor. In this one, we create a 6-input logic AND gate with high dynamic range. But if you use the traditional layered logic gate design methods, we potentially can using 10 transcription factors to achieve this. But here we're using 1 transcription factor. So this means that we can use this split intron and split intin enabled genetic circuit design approach that can have the potential to increase the signal processing capacity of a single transcription factor by 10-fold. And this is what is not limited. And you can use the same approach to build more complex logic gates by means that we can use less amount of genetic content to achieve more complex things. And that is one to reduce the genetic footprint or potentially burden of this circuit design. Especially RNA levels, RNA turnaround is very fast and also have less burden on cells. So that's come to the end of my talk. I'll finish that by summarize that what we do in my lab. As Professor Moon mentioned, in 2022, I moved to Zhejiang University I spent 15 years in the UK. But the research, what I have been working on is to synthetic genetic circuit design and enabling tools development. And we develop these tools and circuits for sensing and computing of multiple cellular environmental signals with applications from biosensing, biotherapy to biomanufacturing. So our research combines both foundational and application um, uh, side of synthetic biology. In foundational side, we developing synthetic biology in labeling tools. For example, we developed these eukaryote-like CRISPR gene activation tools in bacteria, and also the orthogonal split introns for RNA processing, orthogonal split introns for simultaneous for scalase assembly of multiple proteins at the post-translational level. And also we design genetic circuits using these different tools. And on the application side, we use these tools and circuits to develop different applications. For example, these synthetic cellular biosensors, we program these bacterial cells that can detect different environmental or disease signals, and also generate a very easy to integrate of the pattern here. And also we program bacterial cells that can recognize and attack these tumors, the cancer cells, and with precisely this is for disease therapy. And also we can use our tools. For example, these CRISPR tools, we can control the nitrogen fixation gene clusters in the soil bacterium, enhance the nitrogen fixation ability to its host plant. Yeah. And we also have collaborations with leading industrial partners in the field. For example, Microsoft in biocomputing, the Huawei in biomaterials, and the US Navy in biosensing. So that's all from me, and it's great to speak here, and thank you all. Yeah. Now hand over to Lauren. Yeah.
Okay, fantastic. So this is amazing. So, as you know, I mean, when I finished my project, Chris Boyd lab, and one of the comments we got from the reviewer was, can you go further? Just one more kind of layer. So we did actually made the 4-input NAND gate, but after that was made, and then we cannot go further because that circuits start to break. So at that time I realized too much burden, all kind of complexity. So we should choose something else. But now I see after 12 years later, I mean, we have the much better method to build the genetic circuit, and including Chris Boyd work, and then this amazing, 6-input circuit. So I'm so glad. And then thank you so much for your amazing contribution to the community. This is absolutely brilliant.
Okay, thank you. Thank you, Professor Wang. Yeah, I'll stop sharing now. Yeah, and I'll hand over to Lauren. Yeah.
Sure.
Right, yeah.
All right. So now the main speaker of today with a longer introduction. Lauren Andrews is an assistant professor and the Marvin and Eva Schulanger Faculty Fellow in the Department of Chemical Engineering at the University of Massachusetts, Amherst. She is an affiliate faculty of the Molecular and Cellular Biology graduate program, the Institute of Applied Science, and the NIH T32 Biotechnology Training Program at UMass. She received her bachelor degree in chemical engineering from Cornell University in 2006, and then completed her PhD in chemical engineering from the University of Colorado Boulder in 2013 as a graduate fellow under the guidance of Ryan Gill. She was a postdoctoral researcher at MIT and the Broad Institute of MIT and Harvard under the guidance of Chris Boyd at MIT and Robert Nicole at Broad before joining UMass Amherst in 2017. So she is my academic sister. Her research group is developing programmable microbes and bacterial consortia using algorithmic design and high-throughput experimental approach. I believe anything related to engineering consortia should be something we need to do in the future, and that she's absolutely doing amazing, important work at this moment. She has received an NSF Career Award the most competitive important junior faculty award in US, and also a Whiting Foundation Fellowship. She is also a UMass Advanced Faculty Fellow to promote faculty equity in STEM. That's, of course, very important topic we should consider and promote. And also Sloan Faculty Fellow to promote pathways to graduate student for students from historically underrepresented the groups. She is a truly rising star, preparing for a tenure package now. And I want to say, if you happen to be a tenure letter writer, I think you will write one of your strongest letters because she is amazing. Lauren, thank you so much for your time today. And please take it away. And it's all yours now.
Thank you so much for the opportunity to present today. Thank you, Taesuk, for organizing this series and wonderful introduction. I am honored to present today. I just want to check, can you all see the shared screen okay?
Perfect.
Yes. Okay, great. And so today I'd like to share with you some of the work from my group at UMass Amherst about programming synthetic bacterial consortia. So we are inspired by the tremendous diversity of microbial communities in nature that work together to carry out really important and complex dynamic processes which support life, the environment, and health. A few examples of these include human microbiomes and consortia that interact with other organisms such as plants. And if we could harness and design and build such consortia, they could be used for many different applications, from biomanufacturing to agriculture and bioremediation to health. However, there are many challenges to engineering synthetic microbial consortia. And the focus of today's talk will be on two of these challenges. So the predictive design of multicellular functions and cell-cell signaling within bacterial consortia. So in order to have coordination of these dynamic processes in synthetic bacterial consortia, they need communication. So the central programming of bacterial consortia, we need bacterial cell-cell communication. And in nature, this is done by various mechanisms using different types of molecules. Among bacteria, quorum sensing is known to be widely used for communication and can be intra-species, so between the same, or interspecies between different bacterial species. And this intercellular communication between bacterial cells allows for division of labor, where there can be specialization of different tasks and also coordination of these dynamic processes. So this is the first aspect. And the next aspect that we'll focus on today is programming dynamic processes, so known as temporal programming. So these processes performed by microbial consortia, they are not static. They have regulatory networks to respond to various cues and follow prescribed series of states, which can be enzymatic or regulatory. And doing this allows for greater functional complexity and specialization. So examples of temporal programs are cell-cell cycles for cell division and just-in-time transcription with sequential enzyme expression in a metabolic pathway. This allows for synchronization of functions, and in engineered bacteria, this also has many advantages. So for example, implementing regulatory checkpoints for different regulatory states or metabolic phases in order to optimize bioproduction. And so we are very interested in being able to predictively design temporal transcriptional logic in bacteria and bacterial consortia. All right, so how do we do this, I'll walk you through the approach that we are using. So we are composing genetic circuits from transcriptional gates. The simplest gate is the NOT gate, as shown here, which operates as a transcriptional signal inverter. It converts a high input signal into a low output, and a low input into a high output. Now, there are many different biological molecules that we could use for genetic circuits or as inverters. Heard some about that already. In this particular work, we are using DNA binding proteins called transcriptional repressors that bind a specific sequence of DNA called its operator within the output promoter. So in the absence of the repressor, the downstream transcriptional output is on, right, is expressed. And when the repressor is expressed, binds the operator and represses transcription. So the output is off. And this input-output relationship for the transcriptional signal processing, known as the response function, can be measured, right, for a NOT gate, for this gate. So the NOT gate has a single input promoter. However, it was shown by Chris Voigt's group that adding a second tandem input promoter can make a transcriptional NOR gate. So now if either input is high, the output is low, and only when both inputs are low, the output is on. We can write this and represent this in the truth table, where 0 represents low transcriptional signal, 1 represents a high transcriptional signal. And this NOR gate is especially useful because it's what's known as functionally complete, which means any logic function can be carried out using only NOR gates wired together, right? And so, wow, right, we know that signals, right, in living cells are not binary. This abstraction to digital logic is useful for scalable predictive genetic circuit design. So to do this circuit design, we use a set of design rules and a design algorithm. Here shown is an example of the signal matching algorithm, which was demonstrated in the Cello design framework published by Chris Voigt's and Doug Densmore's groups. So not all NOT gates have an identical response function, and the algorithm calculates signal processing using the response functions to predict signaling, and also applies design rules like thresholds to identify allowable genetic circuit designs, meaning those predicted to be functional in living cells. And this was shown to be a quite powerful approach with high predictive accuracy in this seminal paper. Now, while I was a postdoc in the Voigt lab, I was interested, right, if we could extend a similar predictive circuit design framework to temporal programming. So for the sake of time, I'm going to summarize the main takeaway points of the approach that we developed. And this is important as we leverage this framework in our recent work in my group. So basically, in order to have prescribed cell states and checkpoints, we needed to have rewritable memory within the genetic circuits. In digital logic from electrical engineering, this is known as sequential logic, and it allows for predictively designing finite state machines. And in this work, we showed we could predictively design sequential logic that was functional in living bacterial cells. Fundamental to this was designing the memory elements, referred to as latches, that could be used within, right, integrated within the genetic circuits. This is a set reset latch, also called an SR latch, which is built from the same repressor-based NOT-NOR gates that I showed you earlier. And by cross-coupling the NOR gates, it introduces positive feedback, which can give rise to bistability and therefore memory. Memory meaning storing a transcriptional state. We presented a design approach using the NOT gate response functions in what are called null clients in the mathematical analysis here, right, in order to do this design. By selecting gates from the library, we can identify latch designs that are predicted to be bistable. So we show that we could predict the bistability and behavior of the latch using the NOT gate response functions in mathematical models, which then specify the phase diagram and regions of bistability, which can exhibit memory. And importantly, we showed that this was functional. These latches were functional in living cells. Further, we show this approach is extensible to larger sequential logic circuits that implemented temporal programs with up to four states. But then, then we noticed there's a problem, right? And I'm hearing about this already today, right? We hit an upper limit, right, in terms of complexity, which was limited by the parts and gates that are available, and toxicity and cellular burden. And this planted a seed in my mind to pursue a new research direction, and also thinking about, right, these sophisticated bacterial consortia that I showed you right at the outset, right, that use specialization of its members. So up to now, everything that I've shown you has been in a single cell. And now in my group, we've been working to develop predictive design of multicellular genetic circuits. So I'd like to present this vision and tell you this story about our work done by my graduate student, Min, to develop predictive temporal programming of bacterial consortia. So we again wanted an approach that was extensible to a prescribed logic function, meaning used an algorithm to design the genetic circuit from a library of characterized circuit components. But now, right, we needed to incorporate cell-cell gates, right? The method for intercellular signaling within the consortia, right? And the consortia are comprised of at least two different genetically engineered bacterial strains. So for these cell-cell gates, we turn to bacterial quorum sensing. We utilized the large set of prior studies on Lexar family quorum sensors, and our work largely builds upon the work from other groups that have studied and engineered quorum sensors. So here we chose five sensors that each contain a Lexar type allosteric regulator that is activated by a homoserine lactone or HSL signal molecule. Importantly, we don't believe this approach is limited to just these 5 intercellular signals. Just to point out, there is a tremendous diversity of Lexar family homologs that have been identified in various bacteria, especially in proteobacteria. So looking ahead, we see this as a key opportunity. So to implement multicellular sequential logic, we saw that we needed 3 components for the genetic circuits. First, we needed these intercellular sensors. Second, we needed biosynthesis of the intercellular signal molecules. And third, in order to have reversible cell-cell communication and cycling between any states, we needed to also have these signal attenuator components as well. So first, we engineered the intercellular sensors using the selected Lexar family regulators. Importantly, we needed to make sure that these are insulated in their DNA design and compatible with the integration into the genetic circuits. We also needed to characterize the sensor response functions, and we used different strength ribosome binding sites in order to tune each sensor. And we did this for all 5 HSL sensors. And these, right, they have low basal activity and high activation of at least 46-fold and up to 608-fold activation by their corresponding HSL intercellular signal. So we knew from the work of other groups that signal crosstalk can be a problem for Lexar type HSL sensors. So we assayed the dose response of each sensor to all 5 HSL signals. For some, they have no signal crosstalk, but for others, right, high concentrations of a non-cognate signal can activate the sensor. And so, right, here is one example where a non-cognate signal can activate a corresponding sensor. We'll hold that thought for now and then come back to it a little bit later. Importantly, if we've quantified the signal crosstalk, we can account for it in the predictions for signaling within the consortia. Next, we needed to have the autonomous production of the intercellular HSL signals. So we used the corresponding LuxI family HSL synthases for each quorum sensing system. And we could characterize the response in much the same way as the gates and sensors I've shown you for these intercellular gates, here using a 21-hour time point. And this characterizes the intercellular gate, which acts as a buffer or amplifier to transmit signals, right, between strains. Now, the third piece, right, that we needed was our reversible cell-cell signaling using signal attenuation. And here, right, we look to classes of enzymes from other bacteria that cleave and degrade HSLs. These are lactanases or acylases, depending on which moiety in the HSL they cleave. These are also enzymes that are prevalent in nature, believed to allow for interspecies signal interference. And we saw this as an opportunity to leverage this diversity to specify which HSL signals would be attenuated within a multicellular genetic circuit. So here I'm showing you just two examples, and they offer this contrast. So this AIIA signal attenuation gate has broad attenuation. All 5 signals are attenuated, whereas this HAC-B signal attenuation gate preferentially, right, attenuates long acyl chain HSL signals. So depending on the substrate specificity of the attenuator, we can use this within the circuit design to determine which intercellular signals are removed. With all these new characterized components for the genetic circuits, we now have the cell-cell gates. And so we next wanted to use them to test the accuracy of designing multicellular genetic circuits in bacterial consortia, and here in E. coli bacterial consortia. So from a specified logic function, we can determine the partitioning of the circuit and assign gates from these libraries and build the corresponding consortia and compare the experimental measurements to the predictions from the signal matching algorithm. So I'll share just a few examples for the sake of time. This first is a 3-input majority off circuit. It has, right, this is the circuit design with the partitioning and the genetic schematic. Then building and assaying this bacterial consortia, we found that it has a very high circuit score of 83. This is the largest, right, the highest circuit score of those for the designs that we tested. And also this exceeds that of single cell circuits for this function. To point out, to assay the synthetic consortia, we grow them in a co-culture suspension and use a constitutive fluorescent protein to identify each strain and then assay them by flow cytometry. Next, we wanted to try something that exceeds what could be designed for a single cell. This is a 3-input even parity generator. So if you enter it into the Cello framework, right, they can't find a suitable circuit design. However, by circuit partitioning, we could build this functional circuit within the consortia. And also has a quite high circuit score of just above 20. And we see From the genetic circuits that we assay, strong agreement between the measured and predicted outputs for each input combination and strain for these engineered consortia. Next, so we wanted to test, right, whether we could implement sequential logic, right? So what I've shown you is combinational logic, whether we could implement sequential logic in the consortia by wiring our memory elements, right, latches, and intercellular gates. And indeed, we found that this was successful, as shown here with just one example. We collaborated with Professor Doug Densmore at Boston University and his former postdoc, now assistant professor at the Joint School of Nanoscience and Nanoengineering, Samuel Oliveira, in order to build a custom microfluidic environment to grow and assay our engineered consortia containing multicellular genetic circuits. And we found that the engineered consortia grow and exhibit the design functionality on the chips. This provides, right, using this approach and this microfluidic platform provides additional data sets to validate the circuit designs in the consortia. All right, lastly, I'll share one example on this topic showing we could extend this approach to bacterial consortia comprised of up to 3 strains so far. All right, and then from here, I'd like to switch gears and revisit one of these challenges that I presented earlier, which is that, right, there is one intercellular sensor that exhibited high signal crosstalk. This is the sensor containing LASR, and here showing that it is activated by its cognate signal and also a non-cognate signal that differs by 2 carbons in the acyl chain and 1 hydroxyl group. And now, right, what we were thinking, right, what we recognized, other studies have shown that directed evolution of LASR could alter its ligand specificity. So here we hypothesize that we could alter LASR specificity by mutating ligand interacting residues. The crystal structure of LASR was available, and we targeted this region of 6 amino acids in the binding pocket. Some, but not all of these residues were known to affect ligand binding, and one in particular was known to have really important hydrogen bonding interaction with the cognate C12 HSL ligand. Okay, so beyond just identifying improved sensor designs, we also sought to determine, right, the comprehensive protein sequence to function relationship for ligand specificity. This had not been done for LASR or any LASR family regulator. And so we decided we built a pooled library of 9,486 sensor variants using an oligo pool to incorporate each set of design protein mutations, building this pooled library, which had 100% coverage of all the designs in that physically constructed pool. And then we utilized a sort-seq approach to quantitatively screen each sensor's response to both the cognate and non-cognate HSO. This work was carried out by 3 of my grad students, shown here, and I'd like to just touch on some of the highlights from this work in a few slides, and there's a lot more in the publication if you're interested. So within the pool of sensors, it contained the single saturation mutagenesis set, which have 120 LASR variants shown here, substituting all 20, right, each of the 20 amino acids at each amino acid position. Based on the frequency of each sensor design in each of the bins from cell sorting, we calculated then the output of each sensor to each HSL, C12 and C14 HSL, from the sort-seq data set, which was 8.5 gigabases of data. So here, for the single mutation set, we see the original biosensor design containing wild type LASR is here, which shows high activation to C12 HSL and also activation to C14 HSL. We defined this upper rectangle here as high specificity for C12 HSL, and we see in this region it contained 16 sensor designs. We then built and assayed each one of these sensors individually, and indeed we see that all of them show greatly improved specificity and elimination of the signal crosstalk. Within this pooled library of sensors, it also contained the LASR variants that contain all combinatorial double mutations. This is 6,000 sensor designs. These were designed by combinatorial saturation mutagenesis for the pairwise mutations of the amino acids. When we then look at this data set and use the same thresholds that we defined for high specificity, we identify 384 sensor designs, each that have a different LASR variant and have high specificity. So we decided 384 is too many to test them all. So we chose just the 10 that have the highest specificity. We built and assayed each one of these sensors, and we see even further improved ligand specificity within this set. Now, when we look at other regions of this distribution, right, we see a few things. We see a large proportion of the LASR variants that mostly lose activation to either HSL. Others maintain high activation for C12 HSL and have varying activation of C14 HSL. And this really interesting population down here, right? It's a small group, but these now have preferential activation by it was originally the non-cognate signal with the introduction of only 2 amino acid substitutions. We built and assayed all 5 of these sensor designs and observed that yes, they do have the greatest activation with C14 HSL, albeit retaining some degree of activation C12 HSL, and if we're having some degree of crosstalk. In total, we assayed 41 sensors individually, and we see strong agreement with the sensor response that's inferred from the pooled screening in the SORT-seq dataset and the results from these individual sensor characterizations, which validates this approach. Now, looking at the full data sets, there's a lot of information that can be gleaned. Skipping ahead here to show you how we could quantify the sequence to function relationship for ligand specificity for the entire combinatorial design space. And then one of the additional new aspects that we could also interrogate with this big data set is the role of protein mutational epistasis in ligand specificity for LASR. These are the non-additive effects of mutations, right, on the specificity. And we found prevalent mutational epistasis across the library. Here I'm showing you the pairwise mutational epistasis. And this really highlights the need for combinatorial protein engineering approaches. Otherwise we wouldn't find the optimal designs. We did also use this data set to train machine learning models that were developed by other groups. That's in the paper. For the sake of time, I'll skip that part here. The last point I want to highlight from this work is whereas it's typically reported that there's a trade-off between sensor specificity and sensitivity for sensors, including Luxar quorum sensors, we did not observe that here for our engineered biosensors. So we assayed the sensor response to the cognate HSL for 27 of the best designs, which contain 1, 2, or 3 amino acid substitutions, and found that to the contrary, some of the sensors with the best the best specificity also have the highest sensitivity. So from the engineer's perspective, this is quite promising, we think, and that there are a lot more directions to pursue on engineering intercellular HSL sensors. I know we're nearing the end and the end of time. I'll touch briefly on one of our ongoing research directions. So in a collaborative project with Doug Densmore's and Sam Oliveira's groups, we're working to extend this framework to programming diverse bacterial consortia and to integrate dynamic models for the genetic circuit, predictions, and design. Towards this goal, we've been working to extend the potential taxonomic range of this approach using HSL intercellular signaling in programmable bacterial consortia. In 2 slides, I wanted to summarize a really neat finding that shows we could successfully create libraries of highly functional HSL sensors for the bacterium Bacillus subtilis, which is a gram-positive bacterium. For those that don't study quorum sensing, this is cool, as firmicutes don't generally use Luxar-type quorum sensing natively, right, to our knowledge. However, here we showed that by composing these new novel synthetic promoters, we could use the Luxar-type regulators to sense HSL signals in B. subtilis. This required, to do this, combinatorial promoter design in which we altered different regions of the promoter, including, importantly, the -16 region and the transcription start region that have been reported to be important for promoter activity in B. subtilis. In this work, we used statistical design of experiments to design a library of combinatorial promoters. And these are the 36 promoter designs, which sample 50% of the combinatorial design space. We assayed each sensor design, and each one contains a different unique synthetic promoter. And then using statistical analysis, we kind of identify the promoter regions and their pairwise interactions that significantly affect basal activity and activation of the sensor. Okay, so I encourage you to check out the paper if you'd like to find out more about this work. We think this is an exciting prospect towards engineering interspecies and interphyla genetic circuits. Lastly, we have also been very interested in extending temporal programming to non-model bacteria, including those for living diagnostics and living therapeutics. In these 3 papers, we studied the probiotic bacterium E. coli Nissl in 1917, and we demonstrated that with strain-specific libraries, we could predictively design genetic circuits and even new circuit designs that can record biochemical concentration. And again, the papers have a lot more detail if you're interested in this topic. All right, so to summarize, I've shown you about how we are enabling predictive programming of bacterial consortia using algorithms and new cell-cell gates for genetic circuit design in synthetic bacterial consortia, and creating customized intercellular sensors using targeted protein engineering. And also how we're working to enable engineering diverse bacteria and consortia to be programmed as well by teaching an old bacterium new tricks, per se, here teaching Bacillus subtilis to sense homoserine lactone intercellular signals, and how we're working to extend these tools from laboratory strains to non-model bacteria. I must acknowledge my team of incredible folks who did this work and made it possible, and also our wonderful collaborators. Of course, acknowledge our funding for this work. Thank you all for your attention and this opportunity, and I look forward to hearing your questions. Thank you.
Fantastic. Oh my goodness, amazing. This is really exciting. So thank you so much. Let me ask Baojun whether he has comment or questions.
Okay. Thank you, Tae-Suk. Yeah. I think it's a great, fantastic talk. Yeah. I was amazed that the complexity of the circuits that you have achieved, and particularly you proposed and also achieved this with the intercell with this use bacterial consortium to achieve this complex logic circuit design. Yeah. Because we know that the one single cell have the burden have some issues here. We can division of labor into multiple cells, multiple species here. So I think that is one of the directions how to scale up genetic circuit design. I think it's a kind of like in parallel with what I talked today. We mainly talked about how within one single cell we can use less genetic content to achieve more complex things, but instead you use that we can divide the labor into multiple cells. Yeah, that's great. And we know that in achieving that, there are several challenges. One is the cell communication. We must have multiple orthogonal cell communication wires. Yeah. And if it's not very good, you can crosstalk or signal attenuation. This stuff will be very difficult. Another is that if we want to program different bacterial cells or species, and this is non-model stuff. How because we know that this circuit works in one bacterial species doesn't necessarily mean it works in another bacterial species. How we can engineer in this universal genetic circuits that work across different bacterial species very important there. We have done some pioneering work on this to calculate some gram-sensing response modules are working in gram-positive bacteria or even in the probiotic strains. Yeah. So I think on this, and I think you did excellent work. Yeah, and I enjoyed the talk very much, and I think it definitely is a very promising direction in the field, and I think it's just the beginning. I think in the future we probably can just put different bacterial species and to make this like soup that you can just off the shelf you can order that, and that's one of our dreams. Yeah. So I think that is my comment. Yeah. But we know that in the future we also need to demonstrate the modularity and the real like the signal complexity in terms of how many different species you can put together. Yeah. So I think that is we still need to demonstrate. Yeah. But we're working on that, isn't it? Yeah. Okay, that's all from me.
So thank you so much for the comment. Do you have any question? If not, I wanted to address the question from audience.
Please to the audience. Yeah.
Okay, so Kenyon actually asked question that's also related to my question, but Baojun also kind of answered his answer. But the question is, what are the most exciting application of genetic circuit in your mind? And then to add my question, so you present beautifully about the complex circuit building, design and analysis. And also, you also mentioned probiotic. So one of the application, I believe. But as you can imagine, we are building circuit for last more than 20 years, but mostly, in the laboratory environment. I start to, actually, let me put it this way. So I built 4-input NAND gate. I was so excited. And then after that, my excitement is decreased significantly because, this 4-input NAND gate is not going to work in the real world environment. So that's why I choose to go to the more simpler circuit, minimize the burden, etc. And then I now do more application-oriented research than the genetic circuit building for the complexity. at the same line, I mean, this question is probably the same question I have. So I just want to hear from you.
Yeah, so right. Thank you. Both, yeah, the comments, questions, perspectives. One is just to follow up on Baojun's comments. I think this is a really exciting time in work on genetic circuits. I mean, I think we are working on one piece, right? But you know, these can be used in combination, yeah, in the future. So I think yeah, it's really exciting prospects on that. In terms of yeah, the most exciting applications, well, I guess it's hard to choose. As I try to frame at the outset, right? Bacterial communities and consortia are prevalent across nature. They do all sorts of really important things, right? So I'm not to say one is, you know, definitively more important than any of the others, right? I think this is just really exciting. We're thinking about some across different potential applications, some related to biomanufacturing, some related, right? So for optimization of making products using engineered bacteria, some applications related to health, as I touched on, with living diagnostics and therapeutics, and then some applications in other kinds of bacterial communities. So we're right thinking about and working on not just Bacillus subtilis, right, a soil bacterium, but others, thinking about rhizosphere plant-associated microbiomes as well. But I think there are a lot of really interesting potential applications, not just those.
Okay, wonderful. So the Canyon actually asked another question. He's asked, I am curious about the sequence function landscape using just two AA positions on the X and Y axis. Is that landscape just one of many landscapes you are optimizing? It seems like there aren't enough dimension there.
I see. Okay, so I guess the way I should frame this as right, there's kind of limits on the size, right? Size of libraries of pooled sensors that we could build. So we kind of at the outset of this, and given that hadn't been done, chose single amino acid substitutions pairwise, right? And then a subset of all of the triple mutations, right? So probably most of you are aware of this. There's this combinatorial explosion. So what I'm showing you are all of the pairwise mutations, because it's kind of easy to kind of wrap your head around that, right? Is one of the 20 amino acids, right, at each amino acid position across this, right? These six consecutive amino acid residues. But the protein is bigger. You could think about designing libraries, extending this to other regions, right? Other regions of the protein. But really the thing to kind of balance with that is as you expand the genetic design space, the protein sequence space, right? How comprehensively you're able to build an assay, right? Each of those designs, build assay sequence. And whether you have the throughput for that. So this is, right, the sequence to function, right, comprehensive landscape. So all pairwise mutations in this, right, for this region of 6 amino acid residues that I'm showing here.
Okay, got it. So let me double check whether we have more questions.
And I think what we think is really interesting is just by introducing 2 amino acid substitutions in this region, right, we can impart quite drastic changes in the ligand binding specificity of these sensors for really similar signal molecules.
Okay, wonderful. So I don't see any question. And then also I see my clock central time 11:10. So probably we want to close and then chat more later. After, stop recording. So thank you all for joining and staying today. We'll meet again on August 8th, Thursday, the same time, the same Zoom link. We'll have Dr. Christina Smolky now at Anthea, and then Dr. Simone Chiavari at Max Planck Institute for Terrestrial Microbiology. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in joining us and chatting with us. I will promote you to the panelists who can speak and show your handsome and pretty faces if you wish. Thank you. I stop recording. Just 1 second.